Okay. Good evening, all of you. So, last study, what are the class completed? What are the topics we are completed? Yes, anyone? What is the last topic? Yes. Yes, are you there? My voice is audible, students? Yes, all of you unmute.
Yes, sir, you are audible.
Please respond. So, what is the last topic? Do you attend last class?
Mycoplasma.
Sir, I have not attended the class, sir.
Okay. Those who are attending the last class, please respond. What is the last class we
Mycoplasma, sir.
Mycoplasma next.
Few separation techniques we have learned, sir.
Yes. So next. So my voice is audible? Yes.
Yes, sir.
Yes, sir. Okay. Today we will discuss according to our syllabus the fungi. So general characters and classification of fungi, right? So before that, what is the fungi? So before going to fungi, what is the algae? Yes, anyone? Algae. Yes, anyone? Yes, anyone respond, please. What is the algae? Yes. What is the study of algae is called as?
Sir, algae are chlorophyll bearing organism.
So what is the difference between the algae and fungi? What is the study of algae? What we call? Yes.
It is algology or phycology.
Phycology, sir. Phycology.
Phycology or algology, right? What is the study of fungus?
Mycology.
It's mycology, sir.
Mycology. Study of fungus. What is the main difference between the algae and fungi?
Algae are chlorophyllous living organism, whereas fungi are non-chlorophyllous and heterotrophic living organisms.
Sure. Are you sure?
Photosynthesis.
So according to our syllabus, there is no algae syllabus. Only we have fungi. Two types of two blocks. It is covered only fungi, right? I think there is no algae.
Yes, sir.
Yes.
Yes, sir.
So,
Fungi 1, Fungi 2.
So we directly go to the, so you some idea about algae, though it is a freshwater algae, marine algae, terrestrial algae,
Blue-green algae.
Cleophyte algae, lithophytes, phalophytes, thermophytes, planktophytes, planktons, benthophytes, epiphytes, epizoic forms, endozoic forms, parasitic algae. These are the some different types of algae. And this is the algae structure. It is the thallus of structure. It is in unicellular form, the motile, unicellular, non-motile. These are the different types of are there. So the cell structure of algae, the two types of the algae cells are the two types, like prokaryotics and eukaryotics. The prokaryotic cells is found in the cyanophysi members. A eukaryotic cells is characterized by the presence of well-organized nucleus, the membrane-bound organs like plastids, mitochondria, Golgi complex. And it is also the algae member cell wall, flagella, and plastids and chromophores, pigments. The pigments is chlorophyll pigments, carotenoids. The carotenoids are divided into two, carotins and xanthophylls, biloproteins and phycobilins, and some storage product. So these are the some regarding about algae. So coming to fungi. So the our syllabus, it is comprised, first of all, the first topic is the general character of fungi and classification of fungi. So the fungi, is it a slide? So what are the general characters of fungi? The fungi is large group of eukaryotic organisms that includes microorganisms like yeast, moles, as well as the more familiar to the mushrooms are called fungi. The fungus, it is nothing but the mushrooms. You know mushrooms? Yes.
Yes, sir.
Please respond.
Yes, sir.
You know mushrooms.
Yes, sir.
So the mushrooms also different types. Some edible mushrooms, some poisonous mushrooms, some medicinal important mushrooms. We will discuss in the latter. So these fungi groups are the large group of the eukaryotic organisms. The algae members are the eukaryotic and prokaryotic. So like it is yeast, moles, and as well as the familiar of the mushrooms are called fungus. It is fungus. It is nothing but mushrooms. The fungi are achlorophyllous and prokaryotic. Some fungi are prokaryotic, thallophytes. They are found in all type of environment, like moisture content or wet content or dry, arid, semi-arid. The fungi members are, they are found in all types of the environment. It is moist soil rich in organic matter, water, air, dead and decaying organisms, and living organisms. The already study of fungi, what we call mycology, right? So Myceli, the scientist name is Myceli, is considered of father of mycology. Who is father of mycology? Yes, Myceli is considered as the father of microbiology, and D. Barry is considered as the father of modern mycology. D. Barry, the scientist name is D. Barry is considered as a father of modern mycology. The normal mycology, who is the father of mycology?
Myceli, and Debary.
And Debary is the father of modern mycology. So, a kingdom of fungus, some heterotrophic organisms. So, the general characters of fungi, it is, the first of all, it is covering occurrence, thallus organization, different forms of the mycelium, what is the cell structure, nutrition, heterothallism and homothallism, and reproduction. These are the general, it is consist of the general characters of the fungi, right? So coming to, you observe the right side. Okay, my slide is visible. Yes.
Yes, sir.
Yes.
Yes, sir.
The fungi is plural of what? The fungus is derived from the Latin what? Fungar. The fungi are achlorophyllous, heterotrophic, eukaryotic thallophytes, right?
Yes, sir.
So, according to Alexopoulos in the 1962, the fungi include nucleated spore-bearing achlorophyllous organisms that generally reproduce sexually and filamentous branched somatic structure are typically surrounded by the cell wall containing cellulose or chitin, sometimes is both also. Okay, so these are this statement, who given this statement? Alexopoulos. The fungi include nucleated spore-bearing achlorophyllous organisms that generally reproduce sexually and the filamentous branched somatic structure are typically surrounded by the cell wall containing cellulose or chitin or both. These statement given by the Alexopoulos. Next, according to Bessie, the scientist Bessie in the 1968, the fungi are chlorophyllous non-vascular plants whose reproductive or vegetative structure do not permit them to be assigned to position among the recognized group of higher plants. This statement given by the Bessie. And so the branch of botany that deals with the fungi is called mycology, and the scientist who is concerned with the fungi is called a mycologist. So in Usmania University, one famous mycologist, you know that mycologist name? Is a retired professor, is 86 years, I think. You know, mycologist, like Telangana mycologist. Is a famous mycologist. You know that name?
No, sir.
Professor Manoharachari. You heard about this name? Professor Manoharachari is a well-known mycologist. His contribution is very, very large. He is from Usmania University, the Department of the Botany. He is retired. He is a Nazi scientist. Right now, he is a Nazi scientist. So, the sir given identify the many, I think 20 plus new fungal species. He wrote in 100 plus books, and the papers is 1,000 plus publications. His publications in 1,000. So he's very big person. He's in Telangana, very, very famous mycologist. So, and next, P.A. M.Sc is known as father of mycology, and E.J. Butler referred to as the father of Indian mycology. E.J. Butler. Debary is the father of modern biology. E.J. Butler referred to as the father of Indian mycology. So, coming to fungi are non-green in color with the capacity to live in all kinds of environments, right? You know, do you understand this word? The fungi are non-green in color with the capacity to live in all kinds of the environments. Any kind of environment they can grow. Okay. So before going to in the fungi general characters, we just compulsory what are the difference between the algae and fungi. Okay, algae and fungi. So these are the table, some important points covered. The algae, the process chlorophyll, which in some of them is masked by the other pigments such as brown algae, red algae. These are the some blue, green algae. So the fungi, there is no chlorophyll, right? The fungi lack chlorophyll means the chlorophyll is absent in the fungi. In the algae, the chlorophyll is present, but in the different pigments like chlorophyll A, chlorophyll B, and these pigments are different colors. Brown algae, red algae. The red algae belongs to the rhodophyceae. Brown algae and yellow color, it is the xanthophyceae. Orange color, carotenoids. These are the some difference between the algae and fungi. The first point. The next is with the help of chlorophyll, they are able to synthesize their own food material and thus are called autotrophic in their mode of nutrition. Do you understand this point? The algae are they are synthesized their own food material, so they are called autotrophic,
like plants.
And in the mode of nutrition is they're the autotrophs. Auto means it is selfly they are preparing the food material. In
Yes, sir.
yes, in fungi, there is no chlorophyll. The fungi are unable to synthesize their own food materials.
Food material.
So they get it ready-made sources either by the living parasites or saprophytes. Okay? The parasites means they are depends on
Another.
Saprophytes means
Which depends on dead and decaying matter, sir.
Decaying matter. For this reason, the fungi are referred to the heterotrophic in their mode of nutrition. Already, I think we studied in the bacteria. Like already, what is the heterotrophy? So, for example,
yes, sir.
We previously discussed. So, the mode of nutrition in the fungi, for example, it is the heterotrophic. Heterotroph means they are these
the one which cannot prepare its own food material and depend on other plants.
Others. So simply the algae, it is the autotrophs. Simply they are prepared to synthesize their own food materials. Okay, right? So next, the algal thallus is, what is the meaning of thallus?
The plant structure, sir. The basic structure of the algae.
Okay. What about others? Any idea? Thallus.
The plant body is not differentiated into roots, shoot. It is called as a thallus-like structure.
Yes. The thallus, it's look like undifferentiated.
Like it is a primitive one, sir.
Yes. There is
without
no
a differentiation of.
So for generally, for example, in generally the plant is there. So it is differentiated the plant parts like stem, roots, bark, flowers, twigs. These are. So that is the entire plant body. So in thallus, there is no roots, there is no leaves. So there is like it is undifferentiated, right? Do you understand? Thallus means?
Yes, sir.
The plant body is not differentiated into the stem and leaves.
Stem, roots.
And there is no true roots in the vascular system. So these all are the thallus. It is like lichens, some liveworts, the algae, fungi. These are the it is consist of the thallus plant body. Okay, here the algal thallus is composed with the true parenchyma cells. The unit of structure and algal thallus is a cell. The fungal thallus, which is termed as the mycelium. Okay?
Yes, sir.
Generally, what we called in the algal body, what we called?
Thallus, sir.
Thallus. The fungal body, what we called?
Mycelium. We call as mycelium, or we can also termed as hyphae, sir.
Ah, yes, that is also hyphae. Right. Good. Yes. Mycelium or hyphae. Hyphae. So we will discuss in the structure of fungi. We will how it is the hyphae, how mycelium. Mycelium means it's look like some buju antaru kadamma. Idea unda buju.
Yes, sir.
Yes, sir.
That is
Group
also
of hyphae, sir. Fungi. Group of fungi.
ఎస్. ఇప్పుడు జనరల్ గా మనము బూజు చూస్తూ ఉంటాం కదా ఇంట్లోనో, అది ఎనీవేర్ సమ్ బ్రెడ్ పైన లేకపోతే ఎక్కడో ఒక బూజు కనిపిస్తుంది, దట్ ఈస్ ఫంగై. ఓకే, దట్ ఈస్ మైసీలియం. ఓకే,
Yes, sir.
సో హియర్ ద ఫంగల్ థాలస్ is termed as the mycelium is not formed by the division and differentiation of the cells but composed of false tissue called as pseudoparenchyma. It is in algae thallus body. It is composed by the true parenchyma. Here, false tissue called as the pseudoparenchyma.
Pseudoparenchyma
is the unit of the structure of fungus thallus. Is not a cell but a hyphae. So here, what we call the unit of structure in algal thallus, thus it is called as
algal
cell. A cell. Here,
yes, sir.
the unit of structure of a fungus thallus, it is not a cell,
Mycelium.
but it is what we call hyphae.
Okay.
Hyphae.
So in algae, what we call cell. Here, hyphae. So the next point is the cell wall is typically composed in the cellulose. The cell wall in the algae cell wall, it is typically composed in the cellulose.
Cellulose, sir.
In this, in the fungi, the cellular hyphae wall is composed. Here it is what we call hyphae wall. It is also cell wall. It is composed in the fungus cellulose, or often it is called chitin.
Yes.
Okay.
Okay.
Do you understand?
Sir, chitin means, sir?
So that is different. That is the some made up of that is reserve food material. So we study in the like carbohydrates, the different types starch. These are the
yes, sir.
reserve food materials. Other reserve food, the cell wall. Suppose, for example, bacterial cell wall is there. It is composed by the some polypeptide, some the chemicals. The all are the some reserve food material. Same like this. The cellulose is different. Its chitin is also different. So the chitin structure it is different. That is a very big story. So it is cellulose.
It is a reserved food material.
Yes.
Okay, sir.
So these are the chitin. What we called in the fungus cellulose or obtained, it is called the chitin. The cell wall composed in the algae members, it is cellulose. The fungal, it is the fungal cell wall or hyphae cell wall composed in the
chitin, sir.
Chitin. The food reserve chiefly is form of starch. These are the some starch related products like cellulose and chitin. So the food reserve is in the form of starch. So the cellulose composed in the cell wall is composed in the cellulose. The food as a reserve is in this. The form is what form is starch.
And starch.
So here the fungi, the reserve food is invariably accumulated in the form of glycogen, not starch. The two difference. So in algae, reserve food material is the starch. In the fungus, glycogen. Right? Clear?
Yes, sir.
So the algae grow in the habitat, the sunlight, where sunlight is present. So do you understand this statement? The algae, how to grow algae? What are the conditions needs algae?
It is required sunlight.
Sunlight. Why it is sunlight required?
Because it is a autotrophic.
Yes, it is a autotrophs. What is the autotrophs means? Why sunlight? It is they are preparing
photosynthesis process. Its own food material.
They are preparing own food material. The source of energy is the sunlight.
Sunlight.
Right? Clear?
Yes.
Yes, sir.
So generally the fungus grown in the dark and dim light.
Yes.
Because here there is no need of sunlight. Because these are not autotrophs. These are not preparing their own food material. These are depends upon others. These are depends upon the food from the dead and decaying organisms. So there is no need of sunlight for the growing of the fungal species. It is a dark place or dim light, they can immediately grow. You understand?
Yes, sir.
Yes, sir.
So next, they grow in water or any damp soil, sometimes as the epiphytes, rarely as the endophytes. What is the meaning of epiphytes?
Plants which
Sir,
grow
the
on
plant
other
which,
plants.
yeah, sir, for the physical support, like that.
What are the example? Any idea?
Mm,
Yes?
no, sir.
Yes?
Mm,
So you are
mosses, sir, mosses, ferns, sir.
no, I also forget this name. Most of the some epiphytes growing on the ficus tree members like murray and neem trees. Some parasites also. The epiphytes means as an organism that grows on the surface of the plant and derives its the moisture and nutrients from the air, rain, water, or from the debris accumulating around it. Okay. Generally, what are the difference between the endophytes?
Endophytes.
Yes.
Live
Endophytes.
within the living plant cells, sir.
Yes. Yes.
Inside. Endo,
Inside the
that
plant
means
body.
it refers to inside the cover, sir.
Yes. Yes. That is
Ah,
right.
sir.
Okay. So here, the algae grow in the water or damp soil. It is marine source or dirty water. It is sometimes epiphytes. You know epiphytes. The mostly cases in the rare cases in the algae as the endophytes. Okay. So coming to fungi, they have grown in the wide variety of habitat as the parasites in the tissue of the plants and animals. Okay. Plants and animals. So the sexual apparatus increase in the complexity from the simple to the higher forms. We will discuss in the life cycle of different type studies. We will discuss. So there is a progressively and gradually simplification and ultimate elimination of the sexual apparatus from the lower to higher fungi. This is also we will study in the type studies. So these are the main difference between the algae and fungi. You got these points?
Yes.
So some similars between the algae and fungi. So the fungi, algae both prefer to live in the moist environment. In fact, one of the benefits algae derive from their symbiotic lichen relationship, the fungi, their ability to survive on land moist environments. Okay. So I forgot one thing. What is the lichens?
So the group of colony of algae and the fungal family together as a symbiotic relationship.
Yes, a lichen is the composite. It's a group of organisms arise from the algae, some cyanobacteria.
Fungi, sir.
Yes.
Yes, sir. Rhizobium bacteria.
Yes.
In the root nodule of...
No, that is different, not rhizobium. Okay. Cyanobacteria, okay, the filamentous multiple fungi species. It is a mutual relationship. The relationship is mutual. Okay. One question: lichens are fungus or algae?
So both living together.
Yes. That is the good answer. These are the both fungus and algae. And symbiotic partnership. The mutual, both organisms are benefited. So here the dominant one is fungus. So most of the lichen characters from the fungus only.
Why, sir?
So that is nature, like nature. We can't
Okay, sir.
use any. So suppose two organisms combined. So most of the lichens, it is most of the lichens look like fungus-like structures, because that is nature. We don't know now.
Okay, sir. Okay. I
Okay.
thought some functions will be there which do by the fungus. That is the reason fungus will be higher in
Many reasons are there, but exactly reason we don't know.
Okay,
Many
sir.
reasons are there. Okay. So the both algae and fungi are thallophytes. The plant body not differentiated in the root, stem, leaves. Both algae and fungi are placed together in the division of thallophyte of cryptogams, with the exception of blue-green algae. Majority of algae and fungi are eukaryotic. Both the fungi and algae are in general examples of organisms possessing haploid nuclei. This means they have only single copy of each chromosome. It is haploid nature. By contrast, diploid organisms such as human and most mammals have 2 copies of each chromosomes. The vascular system is absent in both the groups, algae and as well as fungi. So both algae and fungi processes cell wall made up of the polysaccharides, chemical nature of the cell wall in algae and fungi. The symbiotic members are present in the both groups, algals with the animals, fungi with the roots of higher plants, between algae and fungi as in the lichens. Both the groups can reproduce by vegetative reproduction, by fragmentation and fission. Both algae and fungi can reproduce asexually by the production of various motile and non-motile spores. The sex organs are naked in both the groups, not protective covering of sex organs in both the groups. These are some similar characters of the algae and fungi. So coming to general character of the fungi. So remaining slides, similarity of algae and fungi.
Yes, sir.
And difference between the algae and fungi. So coming to now general characters. What are the general characters? Some of the most important characters of fungi are as follows: occurrence, thallus organization, different forms of mycelium. The fourth one is the cell structure. Fifth one is the nutrition. Sixth one is the heterothallism and homothallism. And seventh one is the reproduction. Based on these characters, the fungi classification are there. The classification is a very big subject. So according to our syllabus, what are the type studies as well as laboratory in the practical as well as in the classroom we covered. So what are the general character of fungi? How many characters are follows? Tell me. Yes.
Occurrences, thallus organization.
Yes, already the slide is there.
Seven characters.
What is the 7 characters?
Occurrences, thallus organization, different form of mycelium, cell structure, nutrition, heterothallism, and homothallism reproduction.
Okay. In your exams, so the question they are the examiner or question paper asking, write an essay general characters of fungi. So the time must and should you have follow these 7 headings must and should follows. Right? The first one is the occurrence. Second one, the thallus organization. Third one is the different forms of mycelium. Fourth one, the cell structure. Fifth one, the nutrition. Sixth one, the heterothallism and homothallism. And seventh one is the reproduction. Right? Compulsory. It is the very, very important question. The general character of the fungi. Compulsory. You follow these main points. You also, in case you are interested, you studied the different ways like like internet source, some books, whatever may be. You can add some more points also. There is no problem. But well labeling, well diagrams, well subheadings, it is must and should. Okay, you will get good marks.
Okay, sir.
So coming to the first point is occurrence. The fungi are cosmopolitan and occurs in the air, water, soil, and the plants and animals. You know, already we discussed any condition of environment, the fungus is grow. There is no need of sunlight. Okay?
Yes, sir.
So they prefer to grow in the warm and humid places. Hence, we keep food in the refrigerator to prevent bacterial and fungal infestation. So under just you see the right side diagram. So all of you, I think this one you saw many times. It is under the tree members, in the nearby the tree trunks. Yes, this fungus. This is commonly seen in the some under the tree trunks in the, it is some mature places, in the shady places. So these are the occurrence. The second one is the thallus organization. Thallus organization. So here the body of fungi is called, yes?
Sir, the body of
Mycelium, sir.
The body of fungi is called
fungus mycelium.
mycelium. So generally, how you buzu, how it is to look like, it is a thread-like structures.
Yes.
So each thread-like slender structure in the mycelium is called hyphae. So here, the singular and plural are there. For example, how to formation of some the web, you know, web.
Yes, sir.
Suppose the singular, the mycelium is singular, what we call hypha. So hyphae, it is web-like structure. So the web-like,
net, like spider.
Yes. Spider, how spider in the some web-like structures, how to design. So
Yes,
that is hyphae. So it is hypha, it is the singular. The web-like structure is hyphae. Okay, hypha, it means singular, only single mycelium. Okay.
Yes.
So you identify in the right side diagram, it is the unicellular thallus. It is only be seen in the east. In the left side one. In the right side one, filamentous thallus. The filamentous like dots in the right, left, it is alternate, na? That is black spots. Filamentous thallus, for example, in the mucor. So the thallus organization, except some unicellular forms like yeast, the fungal bodies thallus called mycelium. The mycelium is interwoven mass of thread-like hyphae. It is a singular hypha. Hyphae may be separated, which cross the wall, and aseparated without cross the wall. Some fungi are dimorphic, that found in the both unicellular. Dimorphic means it is the both the nature. The unicellular, both unicellular and mycelial forms. It's the mycelial forms. The example is
filamentous
albicans. The thallus may be two types. How many types? Unicellular and
filamentous.
Unicellular example is yeast. In the filamentous, the mucor. So coming to unicellular thallus. In some of the lower fungi, thallus is more or less as a spherical single-celled structure. At the time of reproduction, it becomes a reproductive unit. Such fungi are called holocarpic fungi. In the unicellular holocarpic forms, the mycelium is absent. Some of the holocarpic fungi producing the bud cells in the succession in the remain attached to one another in a chain. The chain of bud cells is referred as the pseudomycelium. These are the regarding about unicellular thallus. So you have what uni means single. The filamentous, it is many. It is the next one is the filamentous thallus. In most of what is the message? Just wait. Someone asked some full screen.
Sir, you can do the slide mode, sir. Slideshow mode.
Right? It is now okay?
Okay.
Someone messaged to me. So full screen. It is okay?
Sir,
Yes, sir, it is clear. Actually, it is clear, sir.
Right side matter కనపడట్లేదు.
Right side matter. Oh.
You don't need right side, no, ma. So what sir is teaching, that is enough. That's a slide.
Yes. There is no need of right side. Okay, actually, I remove that one. Removed. So, coming to chalo. Filamentous thallus.
Yes, sir.
So in most of the true fungi thallus is the filamentous. You know filamentous already. In the previous slides you saw the how filaments. How to single. So the filamentous composed of hyphae. It is loosely arranged hyphae or collectively forms a network known as mycelium. Network means it is web-like. Okay. What is the meaning of web? It is number of connections together. Number of connection. The web means?
Yes, sir.
Number of connections.
Number of connections. So that what we called it is web or network. So here same like this mycelium. Each hypha may vary in the shapes and some hypha different shapes and different types. In the depends upon the fungi. Okay. So each fungi hypha it is totally different in size and shapes. And also the branching of hyphae is dichotomous. What is the meaning of dichotomous?
Branching into two, sir.
Yes,
each branches into two.
Di means two. So, okay, perfect. On the basis of presence or absence of septa of the hyphae of mycelial fungi are the two types. What are the two types? Non-septate or aseptate hyphae. The second one is the
septate
hyphae. So, what are the conditions of non-septate hyphae? The mycelium contains numerous nuclei. The septate hyphae are septate and hypal segments may contain one or two or more nuclei. In the non-septate hyphae, only it contains numerous.
Multi-nucleus. Lots of them.
And also a common mass of cytoplasm without cross wall in the hyphae. Example is oomycetes and zygomycetes, such as the condition is known as. So this one in the type studies we will detail. So what is the oomycetes? You suddenly don't confuse. What is the oomycetes? What is the zygomycetes? You don't confuse. The classification of fungi. What is the oomycetes? What is the zygomycetes? We will discuss in the clearly. Okay, just you leave it. Just example is oomycetes, zygomycetes. That's enough. Okay. So.
Sir, cross wall without cross wall. What do you mean by cross wall, sir?
So cross wall.
Yes, sir.
So the cross wall in the detailed structure of in the hypomycelium, one wall is there. So how to say?
Sir, just like the nodes, what do you call? Is that so? In plants, we have nodes, right? So similarly, the septa which differentiate two cells.
No.
Is that the cross wall?
No. So the cross wall means some differentiate the mycelium parts. So we will let you know in the next slide.
Like a pores, sir?
Yes, parts. It is like
Pores.
parts. Yes.
Not pores. Not
Not
pores.
Not,
It is
not
like
pores. It is parts. Like parts. It is the difference between the parts. The cross wall. For example, the cross road. Like how.
Okay,
The
to divide the structure.
to divide.
Yes.
Yes.
Yes. Yes.
Okay,
So
sir. Okay.
that's the that's okay. That is the cross
Okay,
wall.
sir.
Okay. So dichomals. So separate hyphae, there is it is
It
the hyphal
contains
segments may contains only one or
more
two or more nuclei.
nuclei.
Example is ascomycotina, basidiomycotina, deuteromycotina, and again this separate hyphae is two types of septa: primary septa, adventitious septa. The primary septa are formed in the association with mitotic or meiotic nuclear division, and they separate the daughter nuclei. These type of septa are formed in the ascomycotina, basidiomycotina, and their asexual cells. These characters we are seen in the asexual reproduction of the basidiomycotina, asexual reproduction in the ascomycotina. So don't ask this one. We will discuss again how formation of septa. What are the character septa? We will discuss in the ascomycotina and basidiomycotina and asexual reproduction. Okay. So next, adventitious septa in another type. The adventitious septa are formed in the absence of mitosis or meiosis. It is the reverse to the primary septa. Okay. There is in primary septa with mitotic and meiotic.
Meiotic, yes, sir.
Here,
Nuclear divisions.
the absence of mitotic and
Mitotic,
meiosis
meiotic,
and
nuclear.
occurs especially in the association with the change of the local concentration of the cytoplasm. These are the found in the lower group of the fungi as a mastigomycotina and zygomycotina. So these are the thallus, filamentous thallus. Then the these are the hyphae or filamentous thallus in the septa. On the basis of the presence or absence of the septa, the hyphae of mycelial fungi are two types: non-septate, aseptate hyphae, or septate hyphae. Again, the septate hyphae two types: primary septa,
primary septa, adventitious
septa.
septa.
So coming to different forms of the mycelium. So just you see the right side slide. Different forms of the mycelium. Prosenchyma, pseudoparenchyma. Generally, we observed in the the transfer section of the dicotyledon leaves. What are the structures? Yes. Tell me. Yes. In case we take any transfer section of the dicotyledon, dicotyledons or monocotyledons, what are the appears after transfer section? The section we are observed under the microscope. What are the parts observed? Yes.
So parenchyma test cells.
The first one is the step by step. First one is epidermis is one or two.
Yes,
depends upon the plants. Then
next cortex, sir.
stomata, not cortex. Stomata, some of the stomata. Then polycide. No. Before polycide, spongy tissue. Spongy, spongy. Some gaps are there in the middle of the spongy tissue. Then after spongy,
vascular bundle, sir.
Parenchyma, vascular bundle. Again below the vascular bundle, again start parenchyma. Again spongy. Some species contain spongy. Sometimes some species absent in the spongy. Directly the occupy the rest of the part occupy the parenchyma. Then epidermis are there. These are the structure of the some general dicot leaf. So here, same like this, the different forms of mycelium. So the structure is leaf to leaf, family to family is different. So generally dicot leaf is same. So what is the difference between the monocot leaf and dicot leaf? Yes. Yes, anyone? Some fleshy leaves.
The parallel venish and reticulate.
Yes. Some of the xerophytes in the different anatomy. Xerophytes. Some hydrophytes is the different anatomy. So same like this, it is the different forms of mycelium. The different forms of mycelium referred as a various shapes and the functions of the mycelium which modified according to the circumstances. These include the following forms. The first one is the fungal tissue. What we call fungal tissue? Placental plectin sorry, plectin. It is also called as the fungal tissue. In a fungal mycelium, the hyphae organized loosely and compactly woven to form a tissue called plectin. What we call plectin. The fungal mycelium hyphae organized is loosely and compactly arranged to form a tissue called plectin. In this, two types of plectin are there. One is prosenchyma or prosoplectin. Prosenchyma or prosoplectin. These fungal tissue hyphae are loosely interwoven, laying more or less parallel to each other. The first one, you observe how it is. It is the parallel to each other. It is look like the monocots. So next one is pseudoparenchyma or paraplacencyma. Pseudoparenchyma. In this, fungal tissue hyphae are commonly interwoven, looking like a parenchyma in cross section. When we'll take the cross section of any leaf material, how it is look like the parenchyma, how it is appears in the round shaped or oval shaped. Same like this pseudoparenchyma. Do you understand? Yes, respond please.
Yes, sir.
Do you understand this pseudoparenchyma? When the cross section of, for example, leaf, how the structure is, it is total round shape. Same like this, it is loosely arranged and more or less parallel to, it is looking like a parenchyma in cross section in the like plants. Do you understand this one? Pseudoparenchyma? Yes. Please
Yes,
respond. Without respond, I can't teach the class because you are all are available, or you are listening TV, my class, or you are watching the TV, or you are talking the others. How to my students are listening my class? How to confirm? Only your response. Please give the response. Okay.
Sir.
Yes, sir.
Yes,
Next
sir.
one is the sclerotia. Sclerotia. These are hard, dormant bodies consist of the compact hyphae protected by the external thickened hyphae. Each chlorothium germinates into a mycelium. On return of favorable condition, it is example of the penicillium. Favorable condition means sufficient water, sufficient nutrition, sufficient, it is humidity condition, it is wet condition. These are the favorable conditions. Okay, these are the sclerotia type. And next one is the rhizomorphous. So you see the diagram in the right side one, the white patches are there. Rhizo. Anyone see this? This is mostly in the winter season and rainy season. It is appear of these structures. Rhizomorphous. They are root-like structures. It compactly interwoven hyphae with the distinct growing tip. They help in the absorption and penetration. What is the meaning of absorption? Penetration. The penetration
Soaking,
word.
sir. Like a soaking.
Okay. It is unfavorable periods. The example is Armillaria melia. Next one is rhizides. These are the all are the what are the forms? Different forms of the mycelium.
Yes, sir.
Rhizides. The fourth one is the rhizides. The rhizides is a short, it is root-like filamentous branch of the thallus, generally formed in tufts at the base of the thallus. These also function as anchoring and absorbing. Example is the rhizophidium and rhizopus. So here observe this diagram: sporangium, columella, sporangiospore. These are the rhizides in the root-like structures. What we call it is rhizides. This is also fungal. So next one is fifth one is Apressoria. It is a terminal. This one, in the white black and white diagram. It is a terminal, simple or lobed, swollen mucilaginous structures. Mucilaginous means it is, mucilaginous.
Slimy, sir.
Yes.
Yes, sir.
Mucilaginous structure of infecting hyphae which adheres to the surface of the host or the other substratum, like it is attached to the other surface, whatever may be, it is rock or it is whatever may be, it is attached to the some surfaces. Okay, like host, it is attached to the surface of the host, other substratum, and helps the penetration of the infection hyphae. Infection hyphae, these are formed by the some parasitic fungi such as powdery mildew and rust diseases. Okay, and Astoria. The next one is the Astoria. You see the diagram, fungal hyphae. This is the Haustorium, plant cell, plasma membrane. These are the plant cell. The Haustoria, it is consists of the plant cells. The Haustorium is an organ that is developed from a hypha, usually performing the function of absorption. They are characteristic of obligate parasites. They vary in shape and may be knob-like or button-shaped. Knob-like, how the diagram, just right side, fungal hypha. This is yellow color, the Haustorium. Just you observe these characters. The shape and maybe it is knob-like or button-shaped, elongated, and finger-like or branched. These characters are you observed in the diagram: knob-like, button-shaped, elongated, finger-like, or branched. Yes, yes or no?
Yes, sir.
Yes, sir.
Yes, sir.
Just you observe, you observe the diagram: knob-like or button-shaped, finger-like, or branched. They secrete some specific enzymes which hydrolyze the protein and carbohydrate of the host plant.
Yes, sir.
What is the meaning of
Okay,
hydrolyze?
sir.
Hydrolyze. You know hydrolyze?
Yes, sir.
Yes, sir.
Yes. So what is the meaning of hydrolyze?
Breaking down, splitting of water, sir.
Dissolving, sir.
Dissolve.
Dissolve. Hydrolyze means the dissolve. So
Yes, sir.
the protein and carbohydrate
Carbohydrate.
of the host of the plant. These are the Hastoria. Next, hypal traps. Hypal traps. The predaceous fungi develop sticky hyphae or network of hypal loops known as hypal traps and snares. Just you see the black and white diagram. Loops, hypal loops known as hypal traps or snares. So just wait, just a
Sir, you are in mute.
Sorry for the disturbance. So what is the hypal traps? You see the diagram in the right side. Hypal traps. The predaceous fungi develops sticky hypae or network of hypal loops known as hypal traps. They help in the capturing the nematodes. You know nematodes? Nematodes.
Those insects, sir.
Yes, the one type of the insects. So next, cell structure. The fungi are eukaryotic organism. What are the structure? How the cell structure in the fungi? The cell wall determines the characteristic shape of the cell. It protects the in everywhere like bacteria, virus. The cell wall. What are the function of cell wall? It protects the cell from the osmotic lysis and also act as the binding site for the sub enzymes. So the most is it is protect from the cell wall. It protects from the cell. Okay. The next point is the chemical composition of the cell wall is not the same in the all fungi. The chemical composition of the cell wall in the not same in the all group of the fungi because different fungi in the different chemical compositions are there. The chitin is the characteristically present in the cell walls of the most dominating fungi. The most highest percent of the chitin is there. It is chitin is the cell wall is composed in the chitin. Most of the fungi. The chitin in fungal cell wall is not strictly identical with animal chitin, and the formula is like C22H54N4 has been suggested for the fungal chitin. So, what say this statement? The chitin is different, the fungal as well as animal. It is different chitin. Some animals also contain chitin, but all the chitins are not same. It is different nature and different carbons, different hydrogens, different nitrogen, different oxygens. Okay. So it is polymer of N-acetyl glucosamine. It is a polymer of N-acetyl glucosamine. So the cytoplasm of hypal tip contains Golgi vesicles called cytosomes, which filled with the cell wall materials. And the nucleus and mitochondria are found in content with endoplasmic reticulum. The nucleus divides by the intercellular mitosis called karyochoresis, where nuclear envelope remain intact during the nuclear division and internal spindle develop. The reserve food is a glycogen and oil also present. Most of the reserve food material is fungi is the glycogen. Some fungal species, it is oil also as a reserve food material. Here, the cell wall category, taxonomic group, representative genera. Suppose cellulose glycogen, Arisiomycetes, Polyspondylum, Dictyostelium, cellulose beta-glucan, Oomycetes, Phytophthora, Pythium, cellulose chitin, hypochondration is and rhizomyces, and chitin chitosan, it is Zygomyces, mucurine, phycomyces, chitin beta-glucan, chytidiomycetes, ascomycetes, deuteromycetes, basidiomycetes. It is the representative genera. It is Allomyces, Neurospora, Aspergillus, Foams, and Polyporus. Mannan beta-glucan, Ascomycetes, Saccharomyces, Candida. So these are the fungal cell. What are the components? There is nucleus, ribosomes, endoplasmic reticulum, mitochondria, lipid infusion, plasma membrane. These are the cell wall. So next, nutrition. The fungi lack chlorophyll, everyone knows. Therefore, they cannot synthesize their own food material. It's coming to the nutrition. Their own food material depend on the how they get nutrition. Fungi are following types. Generally, these are the heterotypes. Heterotypes means they
Cannot.
depends others, their food purpose, the food getting from the other organisms. Okay, that one is a three types like saprophytes, parasitic, predaceous, symbiotic, mycorrhizous, and these are the types of nutrition types of in the fungi. So in this first one is the saprotropes. They obtain food from the dead and decaying organic matter. They secrete digestive enzymes to the outside, which digest the substratum and then absorb the nutrients from the dead and decaying matter. Example is Mucor, Agaricus, Rhizopus. So these are the what we call is the type is saprotropes. So how they are look like the saprophytes in the first one. The next one, parasitic. They obtain food from the living organisms. They may be facultative or obligative. The facultative parasite grow on the variety of tissues. Example, Gustiloga. The obligate parasite grow only upon the suitable host. Example, downy mildews. The parasitic fungi that grow on the surface of the host cells and absorb the food through the hostoria are called ectoparasites or ectophytic parasites. Example is Mucor, Ericepae. Mucor and Ericepae. When parasitic fungi grow inside of the host, tissue are called endoparasites or endophytic parasites. Example, Pythium and Puccinia. So next third type is predaceous. Predaceous fungi trap in the nematode. Some of the soil fungi develop ring-like noses to trap the annelids nematodes. Example is Arthrobotrys, Zoophagous, Dactylella. These are the example of predaceous. So next,
they are like predators, no sir?
Yes, yes.
Thank you, sir.
Symbiotic. You know symbiotic. What is the symbiotic relation?
It's a mutual.
Where
both
organisms are
getting benefits.
Yes. Both organisms are getting the benefited. That is the mutual benefit. They are lives in the mutualistic relationship. The mutualistic means it is mutual relationship. They are both the organisms in this symbiotic benefited with another organism by which both are the benefited. There are two common examples, lichens and mycorrhiza. So lichens are symbiotic association between fungi and algae. Okay, the fungal partner is member of Ascomycetes or Basidiomycetes that provides water and nutrients, while the algal partner is the green alga or cyanobacteria. They prepare food by photosynthesis. This is lucky person, lichen, right?
Yes, sir.
Yes.
Yes, sir. It is a mutual understanding. I give you something, you give me something, like no.
But you don't give anything. Just you taken from others, the both sides. Okay. So lichens is a very good. Okay. So in India, the research of lichens is very, very, very low. So
Yes,
some of the researchers, they are now starting the lichens, but it is good. It is a good subject. It is they are good, some medicinal values. They are good. The lichens. So identification of lichens is very, very difficult. Sometimes look like this is a fungus. You consider the lichen. So there is no lichenologist in India. Very less people. So IICT, some of the scientists started in the lichen. So they collect the lichen. First of all, the identification of lichens is very, very difficult. Okay. So, but it is good subject in the lichens. So it is some of the, my friend did my PhD in the lichens from IICT. Now he is a postdoc in the Taiwan. So in case
lichens are also indicators of atmospheric, whether it's a pollution or not. So that is one more thing.
Not only lichen, plants also some pollution indicators are there. So
yes,
but
but specifically lichen, they don't grow in polluted environment.
Yes, yes. So everywhere polluted, no, India. Like Ganga River.
That is why they don't thrive in the cities, but they do thrive in
they
villages.
do in the like villages, some good places, some wildlife, the forest, and some.
Yes, that is what outskirts of the hustle bustle of the.
Okay. So these are the the lichens, the fungal partner members of Ascomycetes, Basidiomycetes, that provides water and nutrition. The algal partner is the green alga or cyanobacteria that prepares the food by photosynthesis, then give to the lichen. Okay. Mycorrhiza. The mycorrhiza are the, it is also some mutual symbiotic association between the soil and fungi. Okay. Soil and fungi. So in the root nodules, it is the soil and bacteria, right? The formation of root nodule, the rhizobacterium is the soil and bacteria. And here the mycorrhizae, it is symbiotic association between the soil and fungi and the roots of the most of the plant species according to the carbohydrate theory. The scientist name is Bojrakman in the 1949 is the plants that grow in the soil different potassium and nitrogen and the high intensity of the light develop the mycorrhizae. And two most common type of mycorrhizae are ectomycorrhizae, ECM, and endomycorrhizae, also known as the arbuscular mycorrhizae. The two groups of is differentiated by the fact that the hyphae of ectomycorrhizal fungi do not penetrate the cell wall of the plant's root cells, while the hyphae of arbuscular mycorrhizal fungi penetrate the cells. So you observe the lichen with the internal structure. That is the algal layer, the fungal hyphae. The hyphae, it is the cross wall. If the center point is the cross wall. Someone asked what is the cross wall? Just you observe this diagram. It is the fungal hyphae. It is the cross mark is there, na? That is the cross wall and attachment of the structure. These are the lichen with the internal structure and plant showing without mycorrhizae. So in the C, it is the width. It is the mycorrhizae without mycorrhizae. These are the two different types of the mycorrhizae and heterothalism and homothalism. So coming to the scientist A.F. Blaxley in the 19th century discovered matting types of genetically distinct strains in the mucor. What is the in short answer? Heterothalism, homothalism is very, very important questions on your exam. Heterothalism and homothalism. So he called fungi with different matting types are called heterothalic. The fungi with different matting types are called heterothalic and fungi without matting types are called homothalic. So here the meiosis, the mitosis, matting, So here, homothallism, mitosis, meiosis, mitosis and matting type, switching, matting. So here, we called fungi with different matting types are called heterothallic, and the fungi without matting types are called homothallic. Nowadays, we call some fungi and algae homothallic if both male and female gametes produced in the same individual can fertilize each other, and heterothallic each other. In heterothallic, if gametes can only be fertilized by the gametes from the another individual of the same species. The heterothallism introduces variation in the species. So we will discuss in the these are the general characters. Just you in your exam in the essay question, just you subheading these 1, 2, 3 points. In each subheading, you cover these points. What is the exactly? What is the heterothallism? What is the you directly write in the heterothallism definition? Bus. Next, homothallism. You directly write in the definition. It is enough. So next, reproduction. In fungi, reproduction may take place by the 3 methods: vegetative, asexual, and sexual. Okay, just you see the this right side diagram. During the sexual and asexual reproduction process, the spores are the essential structures. These are the very important in general knowledge question also. Many times asked in the group 2 question in the GS. The fungi reproduction in spores, something, there are some fruits, there are something, but here the fungi, the formation of reproduction organs is the process is the spores only. Finally, what produce spores. The sexual and asexual reproduction processes, spores, that are essential structures. The spores formed after the meiosis are called meiospores. After the meiosis, what we called?
Meiospores, sir.
Meiospores. Example, ascospores. The ascospores we are seen in the ascomycetes members like penicillium, that we will discuss in the type studies. How they are formation of ascospores, how they are release of ascospores. So, and basidiospores and sporangiospores. The spores formed after meiosis are called meiospores. The examples of ascospores, basidiospores, and sporangiospores. And those resulting from the mitosis. After mitosis called mitospores. The answer is, it is contained in the question only. Okay. Mitospores. It is mitosis. Mitospore. Example, mitospores, zoospores, aplanospores, conidia, and euridiospores. Yes, clear?
Yes,
after
sir.
Meiosis, meiospores. After mitosis, mitospores. The examples of mitospores:
Zoospores, aplanospores, conidia, eurydospores.
So meiospores, example, ascospores, basidiospores, sporangiospores. So these are the you see the diagrams, the right side. Next, the diploid body produced as a result of sexual fusion is known as zygote. How formation of zygote? The diploid body produced diploid body. Haploid means one set of chromosomes. Diploid body means two sets of chromosomes produced as a result of sexual fusion. The fusion means generally in plants, in the reproduction of angiosperm plants, two gametes are fusion of two gametes
coming together, form the zygote.
Here, the diploid body produced as a result of sexual fusion is known as the zygote, which is the lower fungi, as termed as the resting spore, oospore, and zygospore. Right? Do you understand?
Yes, sir.
The processes are almost all similar, but the names is different. Resting spore, oospore, zygospore. These three names also in the different fungal species. Depends on the fungal species. Some of the fungal species, they release the what we call the zoospores. Some fungal species, oospores, some resting spores. In higher fungi, the zygote is represented as a diploid nucleus produced in a cell, ascus or basidium. This diploid nucleus, after undergoing meiosis, results in the formation of applied nuclei, serving as a center for the applied sexual spores called ascospores and basidiospores. So these are the right side. You see the fragmentation. This is the like vegetative reproduction. These are the reproduction in the spores. So coming to the vegetative reproduction. In this type of reproduction, the part of mycelium separate and forms the new individual. The various methods of vegetative reproduction are in this many vegetative reproduction ways are there. That is the first one is the fragmentation, fission, budding, sclerotia, rhizomorphous. These are the vegetative reproduction. In this type of reproduction, a part of mycelium, it is very simple. So this is the fragmentation. This is the entire mycelium. It is what happening in the vegetative. It is cut off the mother cell to, it is in the already cutted part, it is developing a new mycelium. That is the fragmentation. The first one, the hyphae break into the small fragments. Each piece, upon the getting suitable conditions, it germinates to form a new mycelium. This is the fragmentation. So fission. So this method involves in the splitting of the cells into two daughter cells by the formation of constriction followed by the cell wall formation. Just you see the right side. It is a vacuole nucleus developing bud. This is the formation of bud. This is the mother cell. The finally it is dividing cell. Finally in the dividing cells. Okay. Already we studied in the bacteria, but in bacteria it is equal daughter cells. So here there is no equal. The method involves the splitting of the cells into the two daughter cells by the formation of the constriction. When after constriction, there are cell wall formation, they are detached to the mother cells, the formation of finally daughter cells. Budding. A small bud formed in the parent cell, which gradually increases in the size. The right side, you see the right side one, increases the size and receives the part of nucleus, a cell wall formed which separates the daughter cell from the parent cell. Right? It is very simple budding. Sclerosis, these are the penetrating bodies formed by the compact masses intervene hyphae. Sclerotia under suitable conditions germinating to form a new individuals, claviceps and sclerotia. Rhizomorphous, you see the diagrams. These are root-like elongated mycelial threads that remain dormant under unfavorable conditions. Under favorable conditions, develop into the new mycelium. So asexual reproduction, it is commonly takes place through the spores, either motile or non-motile, and form a specialized part of mycelium. The various type of zoospores, spores are there. The zoospores in the asexual, the spores are formed in the asexual reproduction. First one is the zoospores. These are commonly found in the lower fungi like Saprolegnia, Pythium. They are nacked spores which after the forming encyst secrete the cell wall and germinate by the germ tube into the thallus. They are equipped with one or two flagellas. These are the zoospores, the right side, and sporangiospores. The sporangiospores are the aplanospores, non-motile and lack flagella. They are formed inside the sporangium. Example is mucor, rhizopus. These spores may be uninucleate or multinucleate and passes two-layered cell wall. The right side one, sporangiospores. Next, very, very important, the conidia. They are produced in the externally and the branched or unbranched hyphal tips, termed as the conidiophores. The conidia produce the conidiophores. The conidia may be formed in the singly or in the chains. The conidial chains may be basipetal, acropetal. What is the meaning of basipetal and acropetally? Yes, you know, already I explained the most of the time basipetally. Yes. All of you there?
Yes, sir.
So,
What is the basipetal and acropetal?
Sir.
Just you search it and go, and the next class I will ask because the time is over. So that's why I not explain the basic petal and acropetal. It is very simple. The basic petal means
Okay,
the
sir.
older one is the base side, the newer one is the upper side.
Upper side.
So
Yes.
it is acropetal,
Yes,
it is reverse. New one is the base side, old one is
In
the
the
upper
inside, in peripheral, sir.
Yes.
Inside to outside and outside to
Yes.
inside.
The community
Outside.
may be unnucleate or multinucleate. These are the more common in the member of the class Deuteromatis. The ODF. They are produced by the fragmentation of hyphae from the apex up to the base. Each cell thus form rounds of and separates the spore just ODF. This one. You see the diagrams in the like brown and black and the white color. So chlamydophores. They are formed by the rounding of and enlargement of the terminal intercalary cells of the hypha. These can be single formed chains. They do not separate from the hyphae but remain viable to germinate under favorable condition. Ascospores. The ascospores are produced inside in the ascus during the optimal condition. This kind of spores specific fungi classified as the ascomycetes. This one will seen in the Penicillium. Iridospores, a thin-walled red summer spore of the rust fungus produced usually on the leaves of stems of grasses. Though Basidiospores, these are the produced by the Basidiomycetes fungi. They are typically each contain one haploid nucleus. The product of meiosis, they are produced by the specialized fungal cells called Basidia. The sexual reproduction we will discuss in next class. These are the general sexual reproduction, but in the depends upon the fungal species, the different sexual reproduction are there, different asexual reproduction are there, different vegetative reproduction are there. Isogamy, heterogamy, gametal contact, heterogamy, gametangential copulation, spermatization, somatogamy. These are the next. These are the after sexual reproduction. We will go for the classification of fungi. Right? This is the today class. Do you understand today class?
Okay, sir.
Yes, sir.
Yes, sir.
Yes, sir.
General characters of the fungi. Right?
Yes, sir.
Yes, sir. Yes, sir.
Okay. Thank you.
Thank you so much, sir.
Thank you, sir.
Thank you,
Thank
sir.
you, sir.